Been a pandemic, I think there would still be a need for this hearing. This discussion, Dr. Ebright had gotten this started back as early as 2003, 2004. Others have commented on the danger of being able to manipulate influenza viruses to be used as either weapons or by accidental release. But I think given that there was this pandemic, that 1 million Americans died, I lost friends. I have good friends to the pandemic. I think we should be curious. I just am perplexed by the lack of curiosity on some as to know, are there any precautions we can take? Are there any kind of government oversight that we could do to try to prevent this from happening? Now, some will say, well, we can't prove it came from a lab. That's in all possibility true, that we can't prove it, but there are arguments to be made and examination of facts to give us an idea of whether it might have come from a lab. And even if we didn't, I think that this could have come from a person in a lab handling a virus, if it was a virus out of nature, and we've discussed that as well. But I do think that we have to get to the truth of the matter of whether or not dangerous research was going on that should have been reviewable. We had a pause of gain-of-function research, but then we had research occurring during the pause that should have gone to this committee, this P3CO committee, and didn't get to the committee. And I think Dr. Ebright's described it well. He says that in Wuhan in 2016, 2018 period, they were constructing novel chimeric SARS-related coronaviruses that combined the spike gene of bat SARS coronavirus with the rest of the genetic information of a SARS-1 related virus, one that was already known to have lethality. And they found that it could efficiently infect human airway cells and exhibited up to a 10,000-fold increase in viral growth. But then when we've asked before, is this gain of function, we get sort of arguments and protestations that this isn't gain of function, as if this is no big deal and the experts looked at this. But as we look farther into this, we find that the experts never looked at this, that it's sort of a select-in kind of program to this committee. It doesn't go looking for dangerous research. It looks at it if you come to them and say, hey, I think I've got gain-of-function research. Do y'all want to look at my research? And so this opting-in aspect of this, but I think it's important that we get to the truth. Was there research going on in Wuhan that was dangerous? Was it funded by the NIH? And should it have gone through this committee process? I think by the definition that they have given us, gain of function, I think I agree with Dr. Esvelt, could be better defined, and particularly if we're going to have oversight on this, we're going to have to figure out what our oversight is going to be. So by all means, moving forward, we need to ask and include the scientists to get a precise definition of what we're talking about if we want to have more oversight. But we have to look back before we can look forward, not so much to assign blame, but to figure out, is it really necessary? Do we need to be having hearings on this? Should we have follow-up hearings? Should we have legislation? And if 1 million people died and there's a chance this came from a lab, I think without question we should. Both sides of the aisle should be looking at this. So I guess my question, I think it's pretty clear, but I'd like to go through everybody, even though Dr. Ebright has said this was gain of function, to each of the three witnesses. Was the research where you take the backbone of a SARS-1 virus that has known lethality and you mix it together with an unknown bat virus S protein genes to create a new virus? Was this gain of function according to the NIH definition, and should it have been reviewed and discussed by this committee that was supposed to prevent dangerous research from going on? We'll start with Dr. Ebright.
So, as you mentioned, the Wuhan Institute of Virology constructed novel chimeric SARS-related coronaviruses that combine the spike gene of one coronavirus with the genetic information of another. They showed that the resulting viruses efficiently infected human airway cells and efficiently replicated in human airway cells. And they showed that the resulting viruses exhibited up to 10,000-fold enhancement of viral growth in lungs and up to 4-fold enhancement of lethality in mice engineered to display human receptors on airway cells. Based on those facts, and they are indeed facts, the research was gain-of-function research of concern subject to the pause and was enhanced potential pandemic pathogen research subject to the P3CO framework. Nevertheless, due to the failure of the NIH to forward the proposals for review, the work was not paused and there was no P3CO review.
Dr. Quay.
So the Wuhan Institute of Virology is unique in the entire world. Before 2019, 65% of all publications on coronaviruses came from that single institution. They're unique for two reasons. For almost a decade, they were going into bat caves throughout China and actually back into Africa as well, 20 visits a year, and bringing these samples back to the laboratory. So on the one hand, they had the largest collection of raw material backbones from nature to then do gain-of-function research on. They trained in Galveston, Texas, and in North Carolina, and were doing experiments, published experiments between 2015 and 2019. And I believe it's the confluence of those two activities, gain of opportunity, bringing things back from bat caves, and gain-of-function research that led to the pandemic.
Dr. Esfeld?
On the list of experiments that you would need to perform in order to learn whether a novel virus could potentially cause a pandemic, you would need to test growth in human primary cells, such as human airway epithelial cells, and you would need to test transmission in a suitable animal model. So the question is, if they were not intending to determine whether a novel recombinant event between these coronaviruses could lead to something that might kill millions of people, then why were they doing it? If there was no chance that it would come up with a result that looked like it was more dangerous, what's the point? What's the scientific hypothesis? So again, whatever you call it, what they were trying to do was identify a biological agent that has a good chance of being able to kill millions of people if released. And they shared the description of what they did, and they shared the genome sequence because they thought that this would make us safer, because they think that knowing which viruses in nature might cause pandemics makes us safer. They did not consider the security risks, and it's worth noting that both USAID and NIH funded those particular coronavirus chimera studies. USAID, to my understanding, has since disavowed those chimeric recombination studies and announced that they will only focus on finding natural pandemic-capable viruses, which is at least a step in the right direction. But again, I would call that gain-of-function. Another reasonable scientist would say, no, that's not gain-of-function because the term is so ill-defined.
Even beyond the term, though, would it be qualified as dangerous research that actually should have gone before this committee, the P3CO committee, and been reviewed?
Well, here is where you come back to the problem of thinking this as a health and safety issue rather than a national security issue. The question is, why are we trying to identify readily accessible agents that could plausibly be used to kill millions and will, as soon as identified, fall into the hands of all of our adversaries, as well as perhaps individual terrorists who would just want to use them? The fundamental principle behind even wanting to do these experiments in the first place is, I think, a fundamental threat to not just national security, but international security. It's just hard to see why you would ever want to do this when you think about the misuse potential. And I haven't seen anyone else publish a numerical model of that.
Now, people have said, well, the closest relative that we found is only 96% identical to COVID-19. This couldn't have come from the lab. They've also mistakenly accused those who say it came from the lab of saying, oh, it came from this particular variant. But I think what people who are saying that this could have come from a lab are saying that there could also be possibly other viruses that are closer that were manipulated, or that the one that is 96% analogous to COVID-19 could have gone through serial cell culture and become COVID-19. I guess I'd like to ask each three of you whether or not the variant that is 96% analogous to COVID-19, could it through serial passage be transformed to COVID-19? Is it possible? Is it so far away that you can't do it experimentally? Could you do it through gene splicing? Could it be done? Or is it something that argues that this couldn't have come from the lab? We'll start with Dr. Ebright.
The closest relatives are more on the order of 97% identical to SARS-CoV-2 in genome than 96%. Viruses with that level of genetic difference cannot rapidly, in the time scale of weeks or months, move from their state into being a proximal progenitor of SARS-CoV-2. However, in the laboratory, those viruses can be combined at will. They can be combined in particular using a method that would be described as constructing a consensus genome virus. To construct a consensus genome virus, one takes the sequences of several related viruses, identifies the most commonly observed nucleotides at each position in these sequences, and then synthesizes the nucleic acid corresponding to the average, if you will, the consensus genome for the group of viruses. This has been done successfully in coronaviruses. This has been done and published a decade ago in coronaviruses. That kind of research could have been done using viruses that are on the order of 96 to 97% identical in their genome sequences to SARS-CoV-2. And with 2 or 3 or more such viruses, genome sequences, one could develop a consensus. And that's just one of a series of potential routes by which one of the known viruses with 96 to 97% identity could, through a laboratory in a relatively short time, be transformed into a progenitor of SARS-CoV-2.
Dr. Quay?
The three sets of viruses that are closest to SARS-2 are one from southern China, RITG13, and a series of Banal from northern Laos. And as indicated, they're probably 1,200 letters different in the whole 30,000 letter alphabet. In nature, that takes approximately 40 years, so the most common ancestor is about 40 years ago, but most of that can be done in a couple days in a laboratory. However, I don't believe we currently have the starting material, the backbone on which SARS-2 was found. I think it's one of the other 21,000 viruses in the database that was taken down at 2 a.m. September 12th, 2019.
So a great deal of information was destroyed by the Chinese.
It was taken offline, not available. I don't know if it was destroyed. But yeah.
Dr. Esfeld?
If a PhD student proposed to take a 30,000 base paired viral genome and attempt to passage it in the laboratory to acquire 1,000 or so mutations, I would say that is not a PhD project. Go do something else. So I concur with Dr. Ebright that the only way that you could get something so divergent would be to computationally design it and synthesize it, which could certainly have been done. From what data set, and again, why? Why would you do such a thing unless you want to know what the ancestral virus was like and whether the ancestral virus was dangerous? There are basic science reasons why you might want to just know where they all came from, but at the end of the day, the reason why this research is of interest to us is the risk of pandemics. And so again, why would you run the tests to determine whether something was pandemic capable? And they certainly ran those on all of the other coronaviruses that they found and thought might be dangerous. So on the other hand, they never published anything like that, right? And presumably they would have. They published their data on the other stuff. So again, this is why I don't think we have enough information to know. But it was definitely not passaged in a lab from something that was 97%.
I agree. And one of the